Hello, everyone. As you know, genomic medicine is at the heart of personalized medicine with the promise of improving the care of cancer patients. So to solve this challenge, we need to tame NGS by proposing solutions, algorithms, and powerful tools capable of quickly and efficiently identifying the actionable information of the tumor genome, as well as the genomic fragilities of each patient. So today we are pre-announcing our new product line dedicated to somatic cancers and allowing to connect biologists and oncologists together on the single tool while keeping all the information, all the genomic information they need. We believe that this integrated and dynamic practice of using genomic information is key to fulfilling the promise of the genomic personalized medicine. So this is why I would like to give you the floor to Professor Pascal Pujol, President of SFMPP, who will share his vision of the evolving needs of medicine genomic in oncology. So Pascal, the audience is yours.
Thank you very much, Nicolas. I really appreciate it. It's a pleasure for me to talk about molecular profiling and how molecular profiling come to the clinical management of cancer patients. You all know that the role of molecular profiling is really increasing in the management of patients, and that a multidisciplinary approach is very important, involving oncologists, geneticists, pathologists, and that it is really a key challenge of using molecular information for that is called precision medicine. Here, I would like to introduce another actor of this molecular tumor board, which is actually the bioinformatics scientist. And I think that this is a new component, but really required for the best interpretation of data and the best decision to for our patients. Precision oncology is allowed by the combination of advances in knowledge, advance in technology, of course, but also advance of target therapies. If you look behind the last 20 years, we start by the first target therapies in the 2000 years. Now we've got more than 100 molecules with approvals in the clinical situation. We have more than 1,000 in development. To give you also an idea, half of the drugs with FDA approval in the year 2018 was target therapies direct against cancer. Also, another example in France, you can see in the right panel that we've got at present time 12 different authorizations for drug temporary. That means that it's getting more and more complex. So we need help to interpret data, but also to translate them in terms of decision. Another point is that there is growing therapies called agnostic therapies for rare mutation in cancer. And again, this obviously advocates for the use of increase implement NGS in practice, but again, it's had complexity. So that's the point. What for a tumor board? The first question we have to address in clinic is what are the correct tests to use in a given situation. The other is the interpretation of data, and we will go back to this. And finally, it's so-called actionability. That means evidence on the one hand, and in the other, what could we do? Is it target therapies with approval? Is there phase 3 studies? Is this an off-label indication, etc.? It became obvious that we have to put around the table oncologists, geneticists, and pathologists. What is new is that we have to invite a guy called a bioinformatician. Why? Because if for the interpretation of a variant, I mean, you need to be very strong at present time. We need to know if the variant is really a likely pathogenic or a pathogenic variant. Also, we have to know the importance of a mutation in a given pathway. Is this a driver, a passenger? Is this a non-somatic mutation for this type of cancer? What is the level of evidence at the literature at this time? And finally, clinical actionability will be helped by the advice of the scientist in terms of drugability, approval, temporary authorization, basket study, etc. Okay. This is a little bit redundant. I'm sorry about that, but again, all this has to be done in a permanent changing landscape of molecular profiling. So we really have to have people that can make a review of literature about everything, actually, not only the interpretation of data, but also in terms of actionability. Okay, this is very simple draft, but it resumes what we are waiting for, interpretation and molecular analysis. And I will end this talk by real life. For the beginning of the year, we have built a tumor board in Paris involving a liberal clinic, cancer center. And we have to be very careful about that. And as you can see here, I mean, we have a lot of oncologists, of course, but we have a lot of pathologists. I mean, we have 5 pathologists at this board. We also have bioinformaticians, and that is very important. This is just an example, again, in the real life. Talking about a 32-year-old woman. I saw this woman 2 years ago. She has a ductal carcinoma, invasive ductal carcinoma, with node 0, grade 2, estrogen receptor positive, estrogen and progesterone receptor positive, HR2 negative, recurrence score pretty low. This woman undergoes surgery, radiotherapy, and hormone therapy. She didn't get chemotherapy. Unfortunately, I saw this woman 2 years after with a liver metastasis. Because of this young age, we did obviously a BRCA2 test, and we find actually nothing but a variant of unknown significance. When I saw this patient with metastasis, I have the tumor board 2 hours after, so I decided to give to ask people to have a look on that. And finally, in between, actually, the variant was reclassified as a likely pathogenic or pathogenic mutation. I have this oncologist this morning. This woman is under PARP inhibitors and is doing well with a good response. So it is very important and for me crucial at this time to have this update in everything. I mean, on pathogenicity, but not only on actionability. It's becoming too complex for a single brain, even if you put together many brains. We need to be supported in the decision and in the real life by bioinformaticians. Well, this is just my conclusion. So, I mean, we have to fill the gap. We don't need people behind computer, but also not only, but also people participate to the brainstorming and to the decision. Thank you very much.
I think this is the turn of GM. Yep.
Hi, I'm Jean-Marc Holder, and I'm co-founder and VP of Business Development for SeqOne Genomics. Now, I first of all wanted to thank Pascal for having given us such a great overview of the challenges facing oncologists when they integrate genomic data into their processes. And I also wanted to thank all of you for having taken the time to find out about SeqOne.
Yes, this is the plan.
Now, we're here today to pre-announce some exciting new functionality we think will be of interest to anyone analyzing genomic data for oncology. But before we do that, I thought I'd just give you a quick overview of SeqOne itself and how we develop our platform and what we're trying to achieve with it. SeqOne was started based on an observation that sequencing was becoming increasingly affordable, and that at the prices that you can sequence patient data at today, no patient should miss out on this valuable and potentially life-saving technology. However, what we saw was that even though the sequencing itself was cheap, the cost and resources needed to analyze the resulting data and make sense of it remained prohibitive, and that was one of the major obstacles to the more rapid adoption of sequencing and genomic analysis in cancer treatment. So we decided to make a platform that would just take away all those obstacles and make it as easy, effective, and accurate as possible to analyze genomic data. So we approached it by doing three things. First of all, we tried to optimize the whole automation of the process by making an end-to-end software platform that would automatically import raw data from the sequencer at one end and go through all the steps from quality control to primary and secondary bioinformatic analysis, interpretation, all the way through to generating a clinical report. And we believe we've created a platform that minimizes the time it takes to process patient data, giving you the best possible turnaround time. This approach has a couple of other benefits. One, it reduces errors, and two, of course, it enhances your traceability, which means that it helps you with your regulatory compliance. The second thing we spent a lot of time on is bioinformatics. Now, we saw that most people were using standard bioinformatic pipelines like GATK. However, when we did our gap analysis about the kinds of variants those pipelines could detect, we saw that there were quite a few of the types of variants that you really absolutely need in cancer that weren't easily detected. Things like TMBs, MSIs, ALU sequences, mid-size deletions, somatic CNVs, and so on and so forth, as well as those signatures such as HRD that are increasingly important in treating cancer. So rather than starting from scratch and reinventing the wheel, what we did is we benchmarked and took the best open source bioinformatic pipelines we could find. But then to those, we added custom modules that we developed ourselves that detect these rare and challenging variants that otherwise would be missed. So we believe SeqOne gives you the most accurate picture of the cancer of a patient and his cancer, and therefore provide a huge resource, information resource you can use to treat them. Okay, now the final thing that we did is we decided to work on user interfaces that make it easy and efficient for biologists or pathologists to analyze genomic data. And we did this by working constantly with a couple of key players in the sector, exchanging ideas, testing out new prototypes, until we had a solution that we really feel addresses all the needs of people analyzing genomic data. Now, looking at more recent trends in the world of cancer, we see a couple of things happening. First of all, patient loads are going up, so we need to enhance the efficiency of all the labs so that they can keep track and keep step with this increasing demand. Secondly, people are analyzing more complex genes. So technologies like capture are emerging as, the dominant force or the increasingly frequently used technology so we can accurately analyze these complex genes. And thirdly, we see people going to larger panels, as Pascal said, either because they want to see more genes to get a better picture of a patient's cancer, or because they simply want to use the same wet lab to address different cancers, making their whole process more efficient. So again, we've built SeqOne in order to do this, and today SeqOne's platform not only analyzes small panels ranging from really small, 5 or 10 gene panels to the big 500 gene TSO 500 from Illumina and similar, but we also allow you to detect many of these complex variants that you really need, as I mentioned before, things like the MSIs and TMBs. And we invite you to check out and compare the performance of our platform to see if we pick up stuff that's interesting to you. Now today, what we want to do is pre-announce the new functionality in the area of therapeutic recommendation and clinical trial association. These new modules will be available for early beta starting now and will be officially launched in September. However, rather than tell you about all this, what I thought would be better would be to show you our product in action. So I'm going to hand over to Samantha Brunel, product specialist at SeqOne, who will take you through a demo of the product, show you these new functions, and then our CEO, Nicolas Philippe, will close off giving you some insights into our future directions and our vision. Thank you very much for listening, and I'll pass you over to Samantha now.
Good afternoon, everyone. My name is Samantha Brunel. I'm a product expert at SeqOne, and today I'm going to introduce the SeqOne platform to you. SeqOne is a cloud-based solution for genomic analysis and clinical interpretation with applications in hereditary disease and cancer. What you can see on your screen now is the dashboard. This is where you can manage different types of projects around both DNA and RNA data obtained from a variety of techniques and wet labs. It all starts with uploading your files to SeqOne. The platform processes raw sequencing files. Once your data are uploaded, you have access to state-of-the-art genomic analysis tools that are readily available as an app catalog, which you can see now. So whether you are interested in germline or somatic variants, these apps detect and annotate all kinds of genomic variations from frequently occurring small variants like SNVs, indels, to CNVs, to less common yet impactful variants such as mid-sized deletions or alloinsertions. Somatic analysis in particular can be performed on tumoral samples alone or paired healthy versus tumoral samples. You can also compute the MSI status of your samples. Once you have analyzed your data, SeqOne proposes useful information about the quality of your sample that enable you to control the performance of your assay. For example, here is the QC report of this sample, where you find details about the number of reads, their size, the percentage of aligned reads, and the average coverage of your different targets of interest. Now you can take a step further and explore the coverage of your genomic regions of interest at the base pair level using the gene coverage tool. In this view, what you have is access to the coverage analysis for each gene of your panel at the exon level. So let's have a look, for example, at the KRAS gene. And here you can see that all the exons which are covered in our design are well covered. All mutations from a patient are accessible in a variant interpretation table, where ACMG pre-classification along with the user's own variant knowledge base can be leveraged to quickly identify relevant mutations. Now, to narrow down the list of candidates, information about the coding consequence, databases, population frequency, as well as cohort frequency, allelic fraction, and depth of coverage can also be used. So for each patient, you can perform an analysis on small variants, CNVs, gene fusions, MSI, and if you want to browse the aggregated results of all these different pipelines together in a single view, and also obtain actionable insight for your patients, there is another approach to this detailed view. And this is actually where the patient-centered view steps in. Here is a patient that has a thyroid gland cancer, and for which I have actually two different samples. One is DNA and one is RNA, and both have been analyzed with their respective workflow. So written here is my indication, and this is what we're going to use here to identify actionable mutations for this specific context. In the different types here, you have the different types of variants that we have identified for this patient that match this indication. So you have small variants like SNVs and indels, CNVs, gene fusions, and splice variants. Let's first have a look at small variants, which is the first step. Here, the exon 2 of the KRAS gene is highlighted as positive, and we have an automatic quality control that checks that the coverage is sufficient on this region to actually look at the results and the variants that are in there. And below what you have is the mutations that we have found within this genomic interval with both the coding consequence, the depth of coverage, and the allelic fraction of this specific variant. So let's say we want to include this mutation in the next step of our analysis here. Notice that we also have identified a gene fusion involving the red gene for this patient, which is an in-frame inversion. So again, if you want to have access to more details about the analysis, you always have the possibility to go back to the comprehensive output of this specific workflow. So let's say we do it here for this gene fusion. This is what the gene fusion viewer looks like. And it provides an overview of the type of fusion that we have identified and its support in terms of reads, as well as annotation at the transcript level for each of the two partners, and a link to external databases such as Cosmic. Down below you have a visualization that allows you to see both genes, both transcripts, and also the transcript that results from the fusion. And also you can locate your two partners at the chromosome level using this circles view. And finally, have a look at the protein that results from the fusion, where the domains which are conserved are highlighted here in color. So going back to our hotspot view here, let's say we also want to select these variants for our patient as well. So now that we have a short list of mutations, let's gather some more information about their actionability. So in this new view, what you have is a summary of the mutations that we've selected before, both in KRAS and in RET, and the list of drugs and clinical trials that match the indication that we have selected, and the mutations that we've selected, where you can check first the drugs which are a complete match to the indication, but also extend the search somehow to find some other informations. So you can collect some information about each of these drugs, namely the description, indications and usage, and also mechanism of action, and choose to add them to your clinical report. So now let's move on to create the clinical report for a patient. So we can have add the name of our patient here, its date of birth, gender, and also the type of tumor. Information about the sample itself with the percentage of tumoral cells, the stage of the cancer, the type of sample, and the date at which it was collected. You can type in your conclusions and then want to select which mutations you want to include in the report based on the information that you have collected from before. And now let's have a look at the final report, which is going to look like this, with information about the patient, who made the analysis, information about the sample and the mutations that were identified, as well as other types of signatures like TMB and MSI status, the actionability, and the conclusion. So this concludes our demonstration. I will now let Nicolas explain you a bit more and tell you about our future projects.
Thank you very much, Sam, for this demo. So we just watched a quick demonstration of our integrated genomic analysis solution and how it makes it possible to identify all actionable genomic variants and signatures. From that, we are capable to take into account the interactions between them to determine the best therapeutic recommendation with clinical associations inside the dynamic medical reports. So I would like to highlight the fact of having the entire process on the SeqOne platform offers several benefits. The first benefit is the fact that it ensures you traceability of the whole process, minimizing human intervention to improve producibility and reduce the risk of human error. And the second benefit, but not the least, is that it gives us the certainty that the bioinformatic analysis and annotation are done using the latest generation of tools to provide the most accurate identification of rare and complex variants in cancer. So, as you can see, the entire process allows us to provide a dynamic way to get the oncologist to the up-to-date, most relevant therapeutic recommendations, as well as geolocated clinical trial matches. So what we have shown represents major benefits for biologists and oncologists both, because as it provides an easily understood view of the patient and with clear feedback to the best therapeutic options. However, there are still many challenges to address in improving patient treatments. The first challenge is speed, the speed with which medical discoveries in the area of cancer are being made. This means that oncologists have to keep track of a huge amount of new information in order to be able to offer patient latest and most effective treatment strategies. The second challenge is that cancer itself is continually evolving, meaning that a static picture of the patient and his cancer is likely to miss vital information. So we feel the answer to both of these challenges is to move to a dynamic collaborative approach to genomic analysis in cancer. This means by bringing together all the experts that can be helpful to the patient in a collaborative way, collaborative forum, where they can share information and collectively decide on the best approach for each patient. And this collaborative approach means that bioinformatics experts and molecular biologist experts can directly discuss cases with oncologists and other medical specialists to apply the latest genomic knowledge and analysis for each patient. To help in this process, SeqOne is implementing something we call the virtual molecular tumor board. It means an extension of our analysis platform, specially designed to facilitate collaborations. This VMTB allows all the specialists to share their thoughts and information on each patient in a structured manner, which presents all the relevant clinical and molecular biology data in just one place. In this environment, all the variants, all the biomarkers, all the signatures are accessible, not just those selected by biologists, but all the mutation included in the initial genomic analysis. In this way, this is a goal to have access to all the detailed genomic data and allows the VMTMB to explore other hypotheses, especially in cases where patients are at a therapeutic dead end. In conclusion, we are totally engaged in providing the best tools for oncologists and biologists. And this model is one step forward to our goal of creating the best personalized medicine platform for clinical routine using the genomic information. So thanks for your attention, and we hope the opportunity
again, and well, thanks all. Thank you. Well, thanks to Samantha. Hello? Am I on or not?
You
okay, super. Right. Well, first of all, thanks to Sam, Pascal, and Nicolas for some very interesting presentations. And as we said just now, there's time for a few questions. So what I'd like you to do, the chat, and please remember to specify who the question is addressed to. All right. Well, we have quite a few questions in already, so I'll start with one. There's a question about hang on, I've lost it in the chat. There's quite a few questions, so I'll have to look back up. All right, are you using specific software to support your decision at the molecular tumor board? And I suppose this would be a question for Nicolas.
Yep. So I think the question if when algorithm we use to associate mutation and therapeutic recommendation, right?
Yes, exactly.
Okay, so we have a partenariat with Molecular Match, and we build our own decisional tree beyond that to integrate rules for each regulatory territory. For example, in France, we follow the recommendation of ESMO S4, and we follow the recommendation of compare-med rules for Belgium territory. Does that answer your question?
Well, it's not my question, but yes, I think it's a great answer. Thank you. Okay, so for the next one, and I guess this one is either for Samantha or Nicolas. I think Samantha, but I'm not sure. The question concerns regulatory issues, and more specifically, whether SeqOne Platform is aiming to be IVDR. So I think there's a question about IVDR specifically, but perhaps more generally about SeqOne Platform's regulatory compliance.
Yeah, okay, I think I can answer that. So indeed, the SeqOne Platform is CE-IVD, and we are in the process of switching over to CE-IVDR at the moment. Yeah, I think that should answer the question. I don't know if there's more to add to this.
No, that's fine.
Okay.
Thank you very much. Okay. So now I have a question for Pascal. Very early in your talk, you said that one of the main challenges was selecting the right test. And I know there's a big discussion now about some people using targeted gene panels and others using much larger pan-cancer panels. And I wanted to know what your thoughts were on the differences, the benefits and disadvantages of those two approaches.
Well, that's a very good question, Jean-Marc, and I think it's actually a burning question because on one hand, I will say it depends on the type of cancer and situation, of course. But my feeling is the more we go and the more we need very large NGS study. For instance, I show you agnostic therapy in rare mutation. Even if it's rare, if you add everything, at the end of the day, it's a lot because we already know that target therapy are very efficient in that kind of patients. Also, well, just 1 week after the ASCO, again, we have so many markers to incorporate. Not in rare cancer, we're talking about breast cancer at diagnostic. Now we should offer BRCA testing for many women at the diagnostic of breast cancer with high risk. So, and I mean, the story is, you know, going, and I think the more we go, the more, fortunately, it's doable to have this large information, and the more it's actionable. So it's the benefit for the patients.
Great, thank you very much. Okay, so the next question is more about SeqOne, and I think it's a question for Nicolas. It's a three-part question. One, what are the data sources of information in SeqOne or used in SeqOne? 2, do you manually curate? And 3, how frequently do you update? So what are the sources? How do you curate? And how frequently are they updated? Nicolas?
Okay, Sam, you take where I take and you complete as you wish.
Yeah, you can start. I will complete if needed.
Okay, so we have a special module in SeqOne which is capable to plug on the laboratory system, which is called the SDS, and from that we are capable to take into account some both clinical metadata like phenotypes and histoimmunochemie component from the tumor, but also some biological component like the manifest and the library preparation specificities, like what kind of panel we have in the input. It's pretty important to measure the coverage of each hotspot and gives the ability to respond to the answer for are we or not a false negative or a true negative hotspot. Other information like part of the data. No, we have a system. So our algorithm is based on an hybrid system, which we use expert input to compute a model. And by the way of, we use also biological control, and we put all this information together to build a model, and we use some rules, some deterministic rules to learn the model, and after that we propose to the users some automatic rules of classification like ACMG or AMP or such to pre-compute the biological interpretation. And the
That's super.
Question. Yep.
Yeah, so I think that actually another question came in, which I believe you answered, which is whether there are classifications available in the platform. Yeah, and also I think on the earlier question, I got a clarification as well. The question is really about the annotation databases that SeqOne is using and the curation and update of those.
Okay, so Samantha, don't hesitate to jump in the conversation if I miss some important information. So for the classification question, we use 2 levels of classification. One classification for the biological part to classify the pathogenic or likely pathogenic or something like that variant, and another classification to relay the therapeutic recommendation for each variant. So we use together, and this is one differentiator in SeqOne. We call tertiary analysis and quaternary analysis together to both classify variant and therapeutic recommendation in one way. And on the resource management, maybe Samantha, you can take this question.
Yes, I don't know if you wanted some examples of the type of databases that we implement. I think Nicolas illustrated the types that were there. So some of them would be for classification and pathogenicity, and some others for the therapeutical associations and clinical trials. And those databases are then leveraged to pre-classify variants using ACMG rules, etc. And those databases, of course, that we use, there is some part of curation in them, in that we always control the data before we add them to the platform. That's true for databases like ClinVar or OMIM, etc. And the frequency of the updates, well, depends on the resource, I would say. We usually do it something like every 3 months. I believe that it depends, again, on also the frequency of update of the resource itself. And upon updating those databases, we usually notify our users that there was a new version released and that it's possibly impacting also the classification of some variants from the latest release. So yes, I don't know, maybe Nicolas, if you want to add.
Great, thank you very much. That was very clear and very helpful. Thank you. Okay, so I actually had we're getting to the end, so I think we'll only have time for a few more questions. And I actually had one of my own for Pascal. And the question is, in these molecular tumor boards, what is the information that's hardest to get and most lacking to help the group come to a decision? I don't know if this is an easy question to answer, but I was just curious as to what the missing ingredient is.
I think that's really the point. And I try to illustrate that we really need to be on a multidisciplinary board with pathologists, with molecular biologists, oncologists, of course, and you. Because if we don't do that, we will miss something, you know, every time we will miss maybe some clue, understanding to treat correctly the patients.
Okay, and a follow-on question, do you find that because the worlds are very different between the bioinformatics, the oncologist,
That's cool.
the biologist,
like
do they speak the same language? Can they communicate?
I like it. I think we can speak the same language. I'm not afraid of that. I mean, that's the opposite. At the interface of a different discipline emerge very good idea. And I think it's important to involve all these components if we want to be very precise.
Super, thank you. There's a question here for Nicolas. And it's a very general question asking how SeqOne uses AI in personalized medicine and how this impacts the role of the human in the equation. So how does SeqOne use AI?
Yes.
And how
okay, I take it. Our general philosophy is not to replace the human interpretation. We want to use IA or machine learning modules, as you want, to help the biologists and the oncologists in the deterministic way. So the game is to aggregate all this information, human interpretation information, annotation information, and laboratory information, and to extract the crucial, the pertinent information for the biologist he can win some time to in his process to give the good information for the reports, medical reports.
Okay. Thank you very much. Okay, I think we have time for one last question, and that's actually a good one because it's sort of a question that everybody on the panel could answer. And the question is, can you explain the workflow of the molecular tumor board tool in more detail? So we don't really have time for a lot of detail, but maybe if one of you could just explain how the system functions and what it adds to the molecular tumor board process in 90 seconds.
Okay. I think we'll have to make another session.
Yes.
To go further. But it's very simple. I mean, we can go ask question before, ask a question about interpretation. During, that's very important. I insist about that because it's sometimes 1 or 2 shots. We have to be very reactive and to put together all these components of the medical decisions. That will be mine.
So it's really interactive. That's the word I'm hearing is interactivity.
Yes. Absolutely.
Okay, very good. All right, well, listen, I think we have about 23 seconds left. So I'd just like to take a few seconds to thank all of our speakers and most importantly, the audience. Thank you for being here. Thanks for asking all these great questions. And again, it was a pleasure being able to listen to all this interesting information. Have a great day and thanks for coming.
Okay,
Bye-bye.
bye.